Now in this video we are going to continue our cytoplasm component. We were seeing the different various structure present in cytoplasm, and we have learned about mesosomes and cytoplasm. Second structure we will talk about is ribosome. What is ribosome? Ribosome, you can see it is present over here. Okay. So ribosome is a ribonucleoprotein. What does mean by ribonucleoprotein? What is ribonucleoprotein? It is made up of rRNA plus protein. Okay. Now it is a place, or it is a organelle which is used for protein synthesis. So this organelle, ribosome, it is something like this. This is largest subunit and this is smaller subunit. When they will come together, when they will combine together, and there is a mRNA strand, they will combine on mRNA strand from where they are going to synthesize protein. Okay. The process of protein synthesis is known as translation. Okay. Ribosomes are very smallest organelle. The only organelle which is present in prokaryotic cell is ribosome. Okay. One more thing, we have studied about prokaryotic cell, that it does not contain any membrane bounded organelle. Obviously, our ribosome is non-membranous. It does not contain any membrane. So, it is the only organelle without any membrane. It is also known as organelle within organelle. This is a name given for ribosome, or it is known as submicroscopic organelle. Why it is known as submicroscopic? Because it is very smaller. It is the smallest organelle that exists in cell. Okay. What is the function? Function is translation, a synthesis of protein from mRNA. Okay. Now it is made up of rRNA and protein. What is the size or density of 70S? Here the 70S, ribosomes are of two types, 70S and 80S. 80S is present in eukaryotic cell. ATS is present in eukaryotic cell, whereas 70S is present in prokaryotic cell. What is 70S and 80S? Let's see. 70 is S. It means it is S here denote sedimentation coefficient. What is sedimentation coefficient? There is an instrument or equipment for determining the mass or a weight of any particle, or a mass or size of any particle. So this equipment is known as ultracentrifuge. Centrifugation is a method done to determine the size or density of any particle. this 70S, here this S is sedimentation coefficient. What is sedimentation? Sedimentation is when anything is under the acceleration or any speed or velocity is given to any particle and it is moving. Okay, it is moving. When it will get settled down, on what speed it will get settled down, okay, that is known as sedimentation coefficient. So here the sedimentation coefficient, or S, is a Swedberg. Okay, this is to determine the particle size or a density. So here this was about a ribosome. Okay, one more thing, it is also known as polysome. Polysome, when we will say polysome, when it is present in a group of 4 to 5 ribosome on single mRNA. When 4 to 5 ribosome are present on a single mRNA, it is known as polysome. Polysome or polyribosome, and or it is also known as argosome. Okay, all the three names are synonymous, and they all mean that there are 2 to 3 or 4 to 5 ribosome present on single mRNA strand like this. Okay, so this is polysome or polyribosome. Thank you.